For presentation number 2, what we're really going to do here is get more into the neurobiology of things. They've been doing a lot of research and we have a pretty good understanding of what happens. This is going to be the slide-based presentation here for part number 2. This is going to be the pathophysiology of Alzheimer's disease. So if you have that picture slide with you, as well as the slide right under that, I'm going to kind of go through the main ideas of what happens in Alzheimer's disease. We know that one of the genes that's really important, one of the chromosomes, is chromosome 21. We all have chromosome 21. Chromosome 21 codes for something called amyloid precursor protein. All of us have amyloid precursor protein. But for those of us who end up getting Alzheimer's disease, something very unique happens to this amyloid precursor protein. If you look at the picture, you can see that you got that APP on the left-hand side. If you're going to get Alzheimer's disease, it appears that APP gets cut or cleaved by beta and gamma secretase. Beta and gamma secretase come in, they cleave amyloid precursor protein, which leads to the formation of something called beta peptide. And beta peptide is the problem. When people have beta peptide, it starts out with just single loose strands in your brain. But then what seems to occur is those strands get longer and they get kind of a bunch of them and they start clustering. And if you look at the picture, they start clustering and aggregating is the word we actually use. And when they aggregate, they actually lead to the formation of amyloid plaques, big spots of plaques in the brain. So the picture kind of shows that. If you go down to the next slide, it actually shows the beta peptides, the amyloid plaques. Your brain recognizes that there's something wrong, that there are plaques there. So your brain actually mounts a response. It has an inflammation response. If you go to the left part of the drawing, with the inflammation, one thing that occurs is microglia comes in, part of your immune system, and it comes in and it just tries to destroy the plaque. But it is unsuccessful in doing it. It cannot do it. It cannot destroy the plaque or reduce the amyloid being formed. Instead, it starts killing a lot of healthy neurons. And it seems to really destroy healthy neurons, especially neurons that have some acetylcholine and glutamate in them. So neurons get destroyed that have acetylcholine, as well as some changes in glutamate. So that's kind of one side of the picture, the microglia gone wrong with the destruction. On the other side of the equation, when the inflammation process starts, chromosome 19 is expressed, and chromosome 19 does something very interesting. It seems for people who have Alzheimer's disease, chromosome 19 leads to the formation of something called apolipoprotein. If you need to look it up, it might be one of the abstracts, E4. Apolipoprotein E4. And when E4 is expressed, something very unique seems to happen to the neuron. The neuron, where it's expressed, seems to crumble and form tangles. So to give you the kind of counterexample of that, for most people, apolipoprotein E2 is expressed. And when that is expressed, that protein actually allows a neuron to maintain its structure. If you've ever watched Star Trek The Next Generation, structural integrity of the ship means the ship is doing fine. If the ship is not doing fine, we have a warp core breach and we're looking at structural integrity problems. That means the ship is going to blow up. So if a neuron has problems with structural integrity, such as the type that happens when apolipoprotein E4 is expressed, the neuron will basically crumble and form these tangles. So we get the plaques and we get the tangles in Alzheimer's disease. So that seems to be the pathology per se. If you go down to the next slide, I would very much encourage you to look at I think it's a 4-minute video. It actually gives a nice depiction of what I actually just talked about with this particular pathology. If you go on to the next slide, the main areas of the brain that this seems to occur in is the limbic system. The hippocampus is there, that's your memory structures. A lot of emotional centers there as well, and you can actually even see the olfactory bulb coming in, which is why smell things are so very important here. So We know a lot about the pathology. I'm actually going to stop this particular recording at this point, and I'm going to pose a question to you. You can start the next one off with. The question is this. We know all this stuff about what happens with the pathophysiology of Alzheimer's disease. And by the way, I should also tell you that apolipoprotein E4 seems to be very related to cholesterol and LDL, the bad kind of cholesterol going on in your body. So the more you have of apolipoprotein E4, the more there seems to be of LDL in there. What I'd like you to do for just a minute or two before you turn on the next recording is basically think that if you were going to try to intervene in this system, where would you do it? Where would you do it? What would you try to do to stop Alzheimer's disease here? Thanks.